Good morning, students. So today in this video I will be continuing with enterotoxins and your Escherichia coli, which we have started. So we have discussed about the general basic characteristics of Enterobacteriaceae and E. coli. So today we will be discussing about the toxins which are basically produced by your E. coli. Now first is your enterotoxin. Now, this particular enterotoxin means the one which causes some gastrointestinal problem. So, this is produced by ETEC, that is your Enterotoxigenic strain of E. coli. And their main activity is that movement of water and ions from the lumen, from the cell to the lumen, right, of the gastrointestinal tract, resulting in watery diarrhea. If you remember, students, we have discussed about cholera, the cholera toxin, which basically oozes out the electrolyte from the cell into the lumen, resulting in watery diarrhea. The same kind of activity is basically done by this enterotoxin. Now, LT, there is one type of toxin, that is your heat labile. Labile to heat, any protein which is labile to heat, binds with a particular receptor which is called as your GM1 receptor, and this GM1 is called as gangliocyte receptor, which is present on the epithelial cell of the small intestine. So they are present. So if I draw the epithelial cell lining, these receptors are present on the epithelial cell lining of the small intestine, and they basically activate an enzyme ADP ribosylate. So they basically activate ADP ribosylate, which stimulates an enzyme, that is your adenylate cyclase, to increase production of cyclic adenosine monophosphate. So ADP ribosylate, which will increase the production of your activate adenyl cyclase, which will convert ATP to cyclic AMP. So cyclic AMP production will increase. So now what will happen when there is an increase in the cyclic AMP? So when there is an increase in the cyclic AMP, it will alter the activity of your sodium chloride transporters, producing ion imbalance. So there will be an ion imbalance because of the cyclic AMP and result in fluid transport into the bowel. So fluid is being transported into the bowel system and you will have watery stool. So here are some of the E. coli like Enteropathogenic E. coli, right, Entero-invasive E. coli, and there are many other E. coli. So these are the mode of entry. Some actually form your intimintin complex, and they basically with the help of actin bundle they actually infect. Some is phagocytose inside the cell. So these are the various mechanisms which are basically being discussed. Now let's forget that, we will be discussing in the later classes. So toxins in E. coli, so there are stable toxin and your labile toxin. So there are two types of toxin. One is your heat stable toxin, another is your heat labile toxin. So this labile toxin in 1956, the experiment in rabbit. So this is being done in the experiment in the rabbit ileal loop, causing outpouring of your fluids. So it is an ileal loop. So in the ileal loop, there is an outpouring of the fluid. So E. coli labile toxin is like a cholera toxin. Why? Because it is having A and B subunit. A is active subunit, B is the binding subunit. So you know that this is your E. coli toxin which we have talked about. So you know that this is having 5 beta subunits. So this is your beta subunit. So 5 beta subunit. So this is your, if I color it, this is your 5 beta subunits, and AB toxin I have talked about, so it will have both A and B. So this is your 5 beta subunit, and this is your 1 alpha subunit. So 5 beta and 1 alpha subunit is there. So these are toxic. This is having toxic enzymatic activity. Okay. So B causes binding to GM1 receptor on intestinal epithelial cell. Now, component A activate to A1 and A2. So A is further cleaved to A1 and A2. Now this A1, the A1 basically activates your adenylate cyclase. So A1 basically activates adenylate cyclase. So A1 activates adenylate cyclase, and this will result in production of cyclic adenosine 5-monophosphate, and this will increase the outflow of water and electrolyte in the gut lumen, causing your diarrhea. Now, toxins of E. coli, that is your stable toxin. So this is your labile toxin which I have talked about, which is an AB toxin. B binds to the ganglioside receptor, whereas A subunit basically activates ADP ribosylate, causing the formation of your cyclic adenosine monophosphate, and that cyclic adenosine monophosphate will bring about the electrolytic imbalance, resulting in imbalance of the electrolyte and outflow of the water into the lumen. So this is your STA and STB. STA act by activation of cyclic guanosine monophosphate. So it is activating CGP rather than CAMP, causing fluid accumulation. It is not imbalance or removal of fluid, it is rather accumulation of fluid in the intestine. E. coli also produce verotoxins, which causes cytotoxics to the vero cells. It all acts like a Shigella dysentery toxin. So Shiga toxin. It looks like Shiga toxin. Now, if we move on to the classification of your enteropathogenic Escherichia coli, so let's discuss. The first one is your enteropathogenic Escherichia coli, okay, EPEC. Next is your enterotoxicogenic Escherichia coli. Next is your enteroinvasive Escherichia coli. Then enterohemorrhagic Escherichia coli. And then enteroaggressive Escherichia coli. So enteropathogenic, enterotoxigenic, enteroinvasive, enterohemorrhagic, enteroaggressive. So we will discuss it one by one. So enteropathogenic Escherichia coli causes diarrheal disease in children. These are the virulence factor O26, O11, produces verotoxin causing effect in the vero cells. Infantile enteritis and upper part of intestine is affected. The brush border lining of the intestine is lost. Intimecin, EPAC adhesion factor. So this intimecin is an adhesion factor with which this E. coli adhere to the cell. And poor hygiene is one of the cause. So symptom is fever, diarrhea, vomiting, nausea, non-bloody stool. So these are some of the symptoms. Now if you go for the laboratory diagnosis, confirm with polyvalent sera. 2 serological group with polyvalent and monovalent sera is there. And there are the presence of HEF2 adherence factor is another important factor. Okay. Next we will move on to your enterotoxicogenic E. coli. They produce both heat stable and heat labile toxin. Adheres to the epithelium of the small intestine, causing nausea, vomiting, loose motion. HN is like a cholera toxin again, causing accumulation of fluid. Adhesive factors are fimbriae-specific receptors. So there are fimbriae-specific receptor in the intestinal CFA, so intestinal cells, and the mortality of the children usually less than 5 years it is motile. So if we differentiate heat stable and heat labile toxin, you will know that this is like cholera toxin, and it basically activates adenylate cyclase. So here guanylate cyclase is activated and it produces cyclic AMP, it produces cyclic GMP. Here secretion of water and ion takes place, here accumulation of water and ion takes place. So these two are totally contradicting and opposite type of toxins. Now the last slide. Enterotoxigenic E. coli causes traveler's diarrhea and usually seen in the water contaminated with human and animal feces. If you go for laboratory diagnosis, demonstration of enterotoxin in LT and ST is required. Sometimes tissue culture test is done, not much, but ELISA can be done very easily, and passive agglutination test is done for confirmation. The traveler's diarrhea can be detected by this treatment or treated, that is doxycycline, trimethoprim, norfloxacin, other fluoroquinones, and prevention is avoid contaminated food and water, right, to prevent this particular organism from being transmitted into the human body. So next we will start with